Dear friends, welcome to Field Crop video series. In this video, I am going to talk about green gram. Green gram is also called as Mungbean popularly in India. It's a self-pollinated and leguminous crop. It's a third most important pulse crop in India, and India accounts about 50 to 55% of the total world production. India ranks first both in area and production. Area under green gram production has got doubled in the past 2 decades at the rate of 2.5% annually. It's a short duration crop grown as sole crop as well as intercrop and multiple cropping system. Importance of green gram: major consumption as food legume. It contains easily digestible protein and rich source of amino acids. It enriches the soil through atmospheric nitrogen fixation. It adds humus to the soil. It acts as cover crop. Sprouted grains are used as salad and breakfast. It is used in preparing a variety of savory and sweet dishes. It is used as palatable fodder and concentrates of milch animals. Origin and distribution of Mungbean. The primary center of origin is India, that is the wild form is Fasciolus aureus. Secondary center of origin is Bihar in India. Distribution of Mungbean is cultivated in Thailand, China, Philippines, Sri Lanka, India, Myanmar, Pakistan, Bangladesh, Indonesia, etc. In India, mainly cultivated in Rajasthan, Maharashtra, Karnataka, Orissa, Bihar, Andhra Pradesh, UP, MP and all. Coming to taxonomical classification, it belongs to kingdom Plantae, division Spermatophyta, subdivision Angiosperm, class Dicotyledonnae, order Leguminosae, family Papilionoideae, tribe Fasciolae, genus Vigna, subgenus Ceratotopis, species Radiata, variety Radiata, and the diploid chromosome number is 2n equal to 2x equal to 22. Coming to nutritive value per 100 grams of green gram, 100 gram contain protein of about 20 to 28%, fat 3.3%, fiber 6%, carbohydrate 51.2%, minerals 3.4%, vitamins 0.3% and moisture content 10.2%. Botanical description: the habit of Mungbean is annual, herbaceous, erect or semi-erect. The roots it follows taproot system provided with nodules for atmospheric nitrogen fixation. The stem is erect or sub-erect, sometimes twining in upper branches, furrowing and branches moderately or sparsely haired. Leaves are trifoliate, entire ovate and rarely lobed with long petiole. Pods. Immature pods are usually green. Mature pods are iron grey or olive grey or snuff brown in color, round, slender with short and moderate pubescence. Dehisced by both dorsal and ventral sutures into two halves, like this. It contains 9 to 16 seeds. Seeds are globular green. Surface has fine wavy ridges. Hilum is white, more or less flat. Germination is apigeal. Coming to floral biology, the inflorescence of Mungbean is axillary raceme carrying 10 to 20 flowers on long peduncles. Corolla is papilionaceous. Petals are 5, sepals also 5. Mungbean is predominantly self-pollinated. Flower is lightly yellowish, olive to yellow in color with 5 sepals and 5 petals. The outermost petal is known as standard and covers 2 free petals known as wings. The innermost two fused petals are known as keels. The 10 anthers are present in diadelphous condition in 9 plus 1 manner. Ovary is superior, long and multiloculed. Flower. Flower is hermaphrodite, zygomorphic, either lightly yellowish olive or olive yellow. The inflorescence is axillary or terminal raceme with 10 to 20 flowers crowded on long peduncle. This is a flower structure. This is calyx, corolla, the 5 petals. Petals are 5 in number. 3 kinds are standard. The outermost bigger petal is standard petal. 2 wings. Wings are just like the moth wings. Then 2 keel petals are adjoining and protruding inside. These are spirally coiled. Androecium, that is male reproductive part. Stamen has got 2 parts, that is anther and filament. This is anther and the long part is filament. Gynoecium is a female reproductive part and it is having stigma, style and ovary, which is shown clearly in this figure. Floral diagram. It is a hermaphrodite consisting 5 calyx, 5 corolla, 9 plus 1 androecium and superior 1 gynoecium. Coming to anthesis and pollination. In Mungbean, pollination occurs at night prior to opening of the flowers. Anthers start dehiscing from 9 am and completely dehisced by 3 pm. Stigma are by then receptive and are thoroughly covered with pollen. Flower open between 6 am and 8 am and remain open till 11 am. Later, they close between 2 am and 4 pm. Pollen shedding takes place long before the petals open. Cleistogamy occur to an extent of 46%. Pollination is affected in the bud stage on the night previous to the opening of the flower. Here, for your kind information, I would like to say the cleistogamy and chasmogamy. Cleistogamy is the always closed flower condition, whereas chasmogamy is after pollination the flower will open. Variations. Variation in seed color may be green, black, brown, yellow colors. For seed surface, either dull or shining green color. Then flower colors are light yellowish, olive or olive yellow. Pod color, ripe pods irony gray, olive gray or snuff brown. Coming to breeding objectives in Mungbean, primemost is high yield, high grain yield, then different maturity durations, resistance to shattering. Different maturity durations are based on the seasons like Kharif, Rabi and summer. For summer seasons, the maturity duration should be short in order to cover the periods of 2 other seasons. Then resistance to shattering in order to harvest it in time and to avoid the grain loss. Then better plant type, that is idiotype. Then resistant to diseases like Mungbean yellow mosaic virus, resistant to pests like pod borer, aphid, whitefly, better quality with protein methionine content, and the other one is better keeping quality after cooking. Then development of synchronous varieties to enable the hybridization. Coming to breeding methods, first one is introduction, that is intercontinental, intracontinental, interstate. In interstate, Pusa bold from IARI to other states, then Pusa by Saki. Selection and hybridization in this, the variety developed are K, ADT2, ADT3, VBN1, VBN2, pure line selection Co1, Payur1, mutation breeding Co4, that is mutant from Co1. Wide hybridization between black gram into green gram. F1 hybrid seeds were small, shrunken, plants were weak and semi-sterile. Reasons for breakdown of hybrids in seedling stage is due to lethality of hybrids at various stages of development and high degree of pollen sterility. Wild relatives with useful genes. In Mungbean, the Vigna radiata variety sublobata has genes resistance against Mungbean yellow mosaic virus, brood cheats and waterlogging. Vigna umbellata has genes resistance to bean fly. Vigna glaberescens is resistant to CLS. Then Vigna candalensis is resistant to drought and calcareous soils. Segregating hybrid population handling methods are pedigree method, bulk method, backcross, single seed descent, selective intermating and multiple crossing. Coming to selfing techniques of crosses, first one is Shiv Shankar methods. It's a method followed in all pulse crops. In this method, on the evening of previous to the day of pollen shedding, transverse cut is made in flower bud with the help of forceps and needle, then facilitates removal of upper portion of corolla like a cap without causing injury to the gynoecium. Anthers are clipped off automatically and remains inside the corolla cap, which is removed. The stigma is then pollinated with the desired pollen on the next morning. In artificial hybridization, first will follow emasculation, that is preferred during 4 to 6 pm in the evening hours of previous day. Then for emasculation, the young bud is kept between thumb and forefinger. Then point of dissecting needle is inserted just under the standard in an oblique position along the top of the bud. The left side of standard and wing petal are pushed outward and held with thumb. The left hand of keel is removed in pieces with forceps. Then pistil and stigma are exposed. Exposed anthers are removed with forceps. For pollination, the staminal column having stamens and anthers intact are selected. This is taken out from freshly opened flowers. Pollens are rubbed against the stigma of the emasculated bud. Reasons for breaking down the hybrids in seedling stage is due to lethality of hybrids at various stages of development and high degree of pollen sterility. Coming to the varieties, first one is Pusa falguni, developed by IARI, then Pusa Dafsali, that is also developed by IARI, then FS68 from Hisar, C20 from IARI. Other varieties are Kopargaon, BR2, Co1, Co2, Co3, G65, Pusa Baisakhi, Pusa 105, China Mung, Selection 4, Pant Mung 2 and 3. Germplasm resources. The germplasm resources are conserved in Asian Vegetable Research and Development Center, Taiwan, then NBPGR IRI at New Delhi, IPGRI at Rome, Italy, then IPRI at Kanpur, then Punjab Agriculture University, Ludhiana. Important diseases of Mungbean are Mungbean yellow mosaic virus, then CLS, that is Cercospora leaf spot, and powdery mildew caused by Ericife polygony. Root and stem rots caused by Rhizoctonia bataticola and Rhizoctonia solani, and rust. Important insect pests are whitefly caused by the Causalis babesia tabasi, hairy caterpillar, semi-looper, pulse beetle, Resistant varieties to pests and diseases. For thrips, G65, M- SML32, SML100. For aphids, Angul 3-6. For pod borer, V2107. For whitefly, ML series like ML1, ML3. For Mungbean yellow mosaic virus, LM214, Mung number 5. For Cercospora leaf spot, P476, ML34, Gujarat 1. Some of the cultivar and their parentage. Jyoti's parentage is L24-2 into Pusa by Sakhi. Sabarmati parentage is PIM 1066 into Pusa by Sakhi. Then Mohini BR2 into type 2. Recommended varieties for Karnataka are K158 both for Kharif and Rabi. Then PS16 China Mung Pusa by Sakhi S4. The duration of Mungbean is almost 70 to 90 days. Thank you. Hope you liked this video and don't forget to subscribe my channel. Thanks once again.